Okay, it's live. We can start, please.
Okay, start.
வணக்கம் நண்பர்களே, தமிழ்நாடு அறிவியல் இயக்கத்தின் PSL கிளை சார்பாக உங்களை வரவேற்பதில் மகிழ்ச்சி அடைகிறேன். தமிழ்நாடு அறிவியல் இயக்கத்தின் அங்கமாகிய PSL கிளையானது கடந்த 3 ஆண்டுகளாக 41 வெகுஜன அறிவியல் சொற்பொழிவுகளை நிகழ்த்திவிட்டு இப்பொழுது 42வது சொற்பொழிவை நிகழ்த்தவிருக்கிறது. இன்று நம்மிடையே முனைவர் மதுரநாயகி துலசிங்கம் இந்த ஆண்டு உடற்கீறியல் அல்லது மருத்துவத்திற்கான நோபல் பரிசு பற்றி உரையாடவிருக்கிறார். PSL நிகழ்ச்சிகளில் தூண்களாக விளங்கும் பேராசிரியர் TR ராமானுஜம் வந்திருக்கிறார். எங்களுடைய இந்த உரை ஆங்கிலத்தில் இருக்கும். இந்த நிகழ்ச்சி யூடியூப் தளத்தில் நேரடியாக ஒளிபரப்பாகிறது என்பதை மகிழ்ச்சியுடன் தெரிவித்துக் கொள்கிறோம். இன்றைய நிகழ்ச்சியானது தமிழ்நாடு அறிவியல் இயக்கத்தை பற்றிய ஒரு சிறிய அறிமுகத்துடன் முனைவர் சேதுராமன் துவங்குவார். அடுத்து முனைவர் பேராசிரியர் கிருஷ்ணசாமி அவர்கள் முனைவர் மதுரநாயகி துலசிங்கம் அவர்களை அறிமுகப்படுத்தி பேசுவார். அதன் பிறகு உரை துவங்கும். உரை முடிந்த பின் கேள்வி பதில் பகுதி இருக்கும். உங்கள் கேள்விகளை சேட் பாக்ஸில் பதிவு செய்யுங்கள். அதில் உங்கள் ஒலிவாங்கியை அதுவரை உங்கள் ஒலிவாங்கியை நிறுத்தி வையுங்கள். PSL என்ற எங்களது குழுவானது தமிழ்நாடு அறிவியல் இயக்கத்தின் அங்கமாகும். இது உயர் அறிவியலில் கவனம் செலுத்துவதற்கென்று துவக்கப்பட்ட குழு. கடந்த 2018ஆம் ஆண்டு இக்குழு துவக்கப்பட்டது. இதன் முதல் சொற்பொழிவு அறிவியலாளர் ஸ்டீபன் ஹாக்கின்ஸ் நினைவு சொற்பொழிவாக நடைபெற்றது. இப்பொழுது நாங்கள் 41 சொற்பொழிவுகள் நிகழ்த்திவிட்டு 42வது சொற்பொழிவில் அடியெடுத்து வைத்திருக்கிறோம். 18 சொற்பொழிவுகள் அரங்க சொற்பொழிவுகளாக நடத்தியிருக்கிறோம். உலக தொற்று ஏற்பட்டவுடன் சொற்பொழிவுகள் இணைய வழி சொற்பொழிவுகளாக உள்ளன. விரைவில் நாங்கள் அரங்க சொற்பொழிவுகளுக்கு திரும்புவோம் என்று நம்புகிறோம். எங்களுடைய சொற்பொழிவுகள் அனைத்தையும் நீங்கள் யூடியூப் இணையதளத்தில் காணொலியாக பதிவேற்றப்பட்டிருக்கிறது. இணையதளத்தின் முகவரியை சேட் பாக்ஸில் போடுகிறோம். ஜூம் தளமானது வழக்கமாக எங்களுக்கு அனுப்பும் எச்சரிக்கை இப்பொழுதும் வந்திருக்கிறது. இரைச்சல் தொந்தரவுகள் போன்றவரை அத்துமீறி நிலைபவர்கள் பற்றிய எச்சரிக்கை இது. பலரது பெயர்கள் அவர்கள் பயன்படுத்தும் கருவியின் பெயர்களாக எங்களுக்கு தெரிகிறது. எனவே எங்களால் உங்களை தொடர்பு கொள்ள முடியாது. உங்கள் அசல் பெயரை உடனடியாக மாற்றுங்கள் என்று கேட்டுக்கொள்கிறோம். உங்கள் கருத்துக்களை சேட் பாக்ஸில் கொடுத்த இணைப்பை சொடுக்கி பதிவு செய்யுங்கள். எங்களுடைய இமெயில் குழுமத்தில் இணைவதற்கு popular_science+subscribe@googlegroups.com என்ற முகவரிக்கு மின்னஞ்சல் அனுப்புங்கள். சேட் பாக்ஸில் கொடுத்த இணைப்பை பயன்படுத்தி எங்களுடைய டெலகிராம் குழுமத்தில் இணையுங்கள். எங்கள் சொற்பொழிவுகள் பற்றிய தகவல் உங்களுக்கு மின்னஞ்சல் வழியாகவும் கைபேசி வழியாகவும் வரும். இப்பொழுது பேராசிரியர் அவர்கள் அறிமுகப்படுத்த சேதுராமன் அவர்கள் தமிழ்நாடு அறிவியல் இயக்கத்தை அறிமுகப்படுத்தார். Let me start it in English informal. So on behalf of Tamil Nadu Science Forum and Popular Science Lecture Group, I extend my warm welcome to you all. Today we are going to have a webinar on Nobel Prize in Physiology and Medicine in 2021. This lecture shall be delivered by Dr. Madhuranayaki Thulasingam. She is from Stockholm, and we have with us the pillar of PSL, Dr. TRG. We see the registration not only from Tamil Nadu but from other parts of India as well, and we want to have this lecture in English. See, after the lecture is over, it shall be permanently in YouTube and many people will see the lecture, and we want to have it in English. We would like to have the brief introduction about Tamil Nadu Science Forum and the speaker. The program is scheduled on this webinar is firstly the Dr. Seduraman shall introduce the Tamil Nadu Science Forum, followed by the introduction of speaker by Dr. Krishnasamy, and the speaker shall speak for 40 to 45 minutes, and we will have a question answer session about another 30 to 45 minutes. Vote of thanks by Mr. Sellakumar, our EC member, and we will try to wind up the session before 1 p.m. PSL is part of Tamil Nadu Science Forum, and it started functioning in 2018. Although Tamil Nadu Science Forum is functioning from 1980 onwards, it extended its activities in various areas. It came into existence to strengthen the activities in the higher science area. We have conducted 41 lectures so far, and this is our 42nd lecture. This lecture will be, the first lecture was held in 2018 as Stephen Hawking Memorial Lecture. PSL used to have its lecture in the hall, and it adapted to virtual meetings during the pandemic period. We hope we may have few more virtual meetings till the pandemic over, and will be returned back to our hall lectures at least in January 2022. We received warning message from Zoom as that spam or hackers intervention may disturb the meeting, and we would like all of you to exhibit your original name. So some of your names are displayed in your device name. The ground rule is all of you should to switch off your mic until the question answer session starts. As the lecture progresses, any question arises from any of the participants, please post your question in chat window. Your questions will be read and answered during the question answer section. During the question answer section, if any of you want to chat directly with the speaker, please raise your hand and we will allow you one by one to and simultaneously read the question from the chat box and chat window, and we shall also be addressed. We would like to get the feedback of this webinar from you and posted a Google form. The link shall be provided in the chat window. Please fill up the form by providing your contact details and your opinion of the program. We'll keep your contact details confidential, and that shall be used only for the communication of our future programs. In the meantime, I request you to provide your email ID in the chat window by choosing me in the private chat. And we also have a Telegram group meant for PSL. To join the Telegram group, we are going to provide a link for the chat window, provided in the chat window. We also have the Google group, so you can send a mail to the Google group. The email ID for the Google group is also be sent to you. With this, let me hand over the mic to Dr. Sethuraman to introduce Tamil Nadu Science Forum. Sethuraman. I think it's not. I saw him. I think he was dropped out of it. SK is part of Tamil Nadu Science Forum. You can introduce the TNSF and the speaker. SK, can you do that?
Okay. Yeah. Thank you, Jen, and the Popular Science Lecture Series. Tamil Nadu Science Forum has been there since the 1980s and been involved in science popularization. Initially, it was more or less in the city of Madras at that time. Then it sort of during the Arivali Yakkam time, it expanded to all the districts in Tamil Nadu. And now it has sort of become well established in almost all the districts. And there are different areas of operation working in the Science Forum. And one of them is the science communication, and that science communication looking at more specifically higher education research. Vijayan, Govindarajan and others in Chennai have started this effort, Popular Science Lecture. And so now I'm happy that we are having the 42nd lecture series in this. So with that, I will just go on to introduce Madhuranayaki Thulasingam. Is a very dynamic person, well-established researcher, and she is a very good human being. She actually did her bachelor's in medical lab technology around 2004, and then she worked for a year in the WHO in a WHO project with now the famous ivermectin. So I really don't know what was the work there, but anyway, now then after that she did her MSc in biotechnology in Mother Teresa University in Kodaikanal, and then she joined me for PhD in Madurai Kamaraj University in structural biology. She did a lot of work on the porin, outer membrane proteins, porin, OMP-F and OMP-C, and she really drove the project on the antibiotic interactions. And then during that, she had opportunities to visit to Spain and various other places. And she then subsequently moved on to Stockholm, where she worked in this Karolinska Institute. And I think she will also tell you that she happened to be in a lab for Nobel laureate protege, or somebody who worked with the Nobel laureate. And I think she also had opportunity to interact directly with David Julius. Maybe she will tell a little more about that incident. And then now, of course, she has moved on after working in the Institute Karolinska for about 8 years. She has moved on to a company that is very, I can't pronounce the name of the company. It is what is
Vironova.
It is Vironova AB. Okay.
Yes.
So yeah. So, but one thing I must tell the Science Forum and all the people is that Madhu, apart from all this, is also an activist, and she was with the students group actively protesting against the various problems that happen in the university. And so I was happy that she was able to complete her PhD and move on. So now it's also a pleasure to invite Madhu to give this talk, and she's an excellent teacher. So I'm sure that it will be nice to hear her. Yeah. So Madhu, please go ahead.
Thank you, SK. It was wonderful to hear from you, and I can relate that. And yeah, it's great. So I will start sharing my screen here. I hope everyone can see my screen now. Can anyone please confirm it?
Yes, we do.
Yeah, thank you. First, I would like to thank Tamil Nadu Science Forum and SK and Mr. Vijayan for giving me this opportunity. I feel very honored to present this, to give this talk today, because as you see, the 41 lectures so far has been given by eminent professors. And I'm having very less experience, and I've been invited by SK, and I really, really thank SK for giving me this opportunity. And I thank Tamil Nadu Science Forum. And when I was during my PhD, I have been involved in Tamil Nadu Science Forum activities in Madurai. And it's a wonderful feeling because we go and reach children in the villages and talk science to them, and then explain science and make them to work on science. And taking science to the public is really a good feeling, because when the people don't know about science and what is happening exactly, and they realize, oh, this is that, and that when you explain to them, and that's a real nice feeling. And I really, really thank SK for giving me that opportunity to do that. So after completing PhD with SK, and I moved to Karolinska, and still I am miles away, and I got this opportunity. And again, I'm very thankful to you, SK, and to Vijayan, as well as to Tamil Nadu Science Forum and all the members in the Science Forum. Today I'm going to talk about this year's Nobel Prize on Physiology or Medicine for the discovery of receptors on which is responsible for sensing our temperature and touch. So I'll go into my presentation here. So this year Nobel Prize has been given to Professor David Julius, who is a professor at University of California, as well as to Professor Ardem Patapoutian, who is the professor at Scripps Research, for the discovery of these two proteins, as you see here. So David Julius for TRPV1, and for Professor Ardem Patapoutian for piezo channels. So this Nobel announcement is actually very, Nobel selection is a very top level secret. So they will inform even to the laureates just a few minutes before the press release. So it's always been a difficult situation the people from Nobel Forum faces, because if the laureates are from some other country, and there is a time difference between Sweden and US, so they have to reach it's in the middle of the night. So like that, there will be a story for most of the laureates, those who are from other country. So in our case, in David Julius' case, they tried, Nobel committee member tried to reach him, but it was middle of the night. And then somehow he could catch hold of his sister-in-law, who lives miles away from his place. And then she was not sure, and she was not sure to give his phone number or not. And they said, oh, we are calling from Nobel Forum, and Okay, and then she talked with David Julius' wife, and then only after a lot of confirmations, they handed the call to David Julius. And as you can see, the picture of him waking up early in the morning, like 2 a.m., and he says that, oh, it's worth waking up early to receive this information, news. And in case of Ardem Patapoutian, it's the same. And they tried, they couldn't reach him directly. And then they called his father, who is 92 years old. So Ardem kept his phone on do not disturb mode. So, but in this mode, only very close relatives can contact him. So then his father, who is 92 years old, who lives in Los Angeles, and he reached Ardem Patapoutian to convey the message. So this is a small story how they received the award in news. And as you all know, the Nobel Prize is given in Stockholm, but specifically Nobel Prize in Physiology or Medicine is given in Karolinska Institute, so where I worked until the first week of September. And there are different universities like Stockholm University, where the committee itself is in a different places. So for example, Stockholm University is responsible for Nobel Prize in chemistry, and for physiology and medicine, it is Karolinska Institute. Yeah, as SK mentioned that my interaction with David Julius. So I visited California in 2019 for a seminar on bioactive lipids, and David was there, and his presentation was amazing with his work. And I was so inspired because I'm a structural biologist with a lot of structural details, and it's a very, very marvelous work he has done. And I was talking with him, and after that, we booked a taxi to airport. And our taxi hasn't arrived, and his taxi arrived before us. And he was saying, "Come on, let's go. It's a very big one. What I'm going to do with a big one?" So we all traveled. I traveled with him to airport, and he was a very humble person, I would say. And he is so much into nature. So how these molecules, how we sense this and everything. So it was a wonderful feeling and unforgettable moment in my life to traveling with the Nobel laureates. I didn't know that at the time he's going to receive a Nobel Prize. Otherwise, I would have clicked a picture with him. I don't have any picture with him at the moment. Yeah, but no problem. He will be coming, hopefully, this December to receive his award to Stockholm. And usually the Nobel laureates will give a Nobel lecture at Karolinska Institute. So hopefully with the corona and restrictions lifted, if it goes fine, so I'll get a chance to meet him this year. So why did they got this Nobel Prize? Is for discovering these two receptors. So these receptors are actually proteins. So in our body, we have myriads of proteins. So each protein has one function. So that is that's a whole mystery. So unraveling this mystery is a basic scientific research. So which protein is doing what function? So this year, Nobel Prize has been awarded to unraveling one of that mystery. That is how we sense the temperature. So this protein, which is present in our body, helps us to sense the temperature. And this protein, piezo channel, which is present in our body, helps to feel that touch. So this is the basic nature mechanism built in our body, which has been unraveled thousands of years later. So it's a great thing, and I will present deep details about this in my forthcoming slides. So let me start with very basic, our senses. So our senses help us to perceive the world around us. So all this information is completely this physical and mechanical information around us, the world, like the vision. I can see, I can see, oh, it's my monitor, it's my curtain. So how I'm interpreting the world by looking at it. So this vision sense is helping. And by smelling, okay, I have my coffee cup, I can smell, yeah, it's a coffee. And I have my water, yeah, it's water. Water doesn't have any smell, but I can but different varieties of perfume, food. So our sense of smell help us to appreciate what it is. And the taste. So I have my coffee, I drink my coffee. It's bitter. I haven't had sugar. So I can taste it. So I can feel and I can perceive and how it is. And hearing, okay, I'm talking, I can hear if someone else talks. So all these physiological, mechanical information is completely depend on our biophysical and biochemical properties of our central nervous system, which is our brain. That gives all the information, all the senses, how we see, smell, taste, and listen, everything. So these mechanisms has been very well established, the molecular mechanism behind these senses, except this somatosensation. So when we Google it, what are our senses? It usually says it is touch. It is not only touch, this somatosensation, it also includes thermosensation. So what is thermosensation? I take a coffee cup and I touch it. It's warm because it's been some time I prepared the coffee, so it's a bit warm now. It's not super hot. So how do I feel? That sense of feeling is thermosensation. And nociception. So if it is a very hot coffee, and if I touch it for too long, it's painful. So that realization, how I'm getting, that is nociception. And then the touch, I can touch the coffee cup. If my child come and touch me, I can touch and feel her skin, and that's a touch, how I can detect it. And the final thing is proprioception. That's very interesting. We never ever thought about this one. It is how our hands and our legs located or oriented with respect to our body. In a simple example, I wanted to touch my head. So then suddenly my hand goes and touches. I can close my eyes and I can touch it. I don't need my eyes to open and then, okay, am I going there or there? I want to stretch my hand there. Yeah, I can do it without looking at up. So how I'm doing that? Because of the proprioception. And a best example is doing exercise, yoga. We do different postures in yoga. We stretch back, front, and everywhere. But we don't look at all the direction. Is it going this side? Is it going that side? No, we just do it. It's because of this sense, proprioception. And playing an instrument like a piano or, violin. So you don't look at all the time once you practice, and then your hands are doing the function without looking at. So this are called proprioception. So how we are doing it? Have you ever thought about that? How I'm touching my nose without looking at, how I'm touching my hand, how I'm walking, what sense is making me? Is it it vision? You can walk casually, but of course not in the highway when the buses are going on. How did we talk about that? There is no, you don't sense, you don't smell. So this is what is more important, is one of the sense which is present in our body, but the molecular mechanism has not been very well established. And the other side of this touch is a pain. If that is a stimuli, and we get an acute pain, and then actually pain is one of our body's protective system. If you don't feel the pain, we will be keep on touching the stimuli, and it causes injury to our tissue. So, but when the stimuli is gone, but still we have pain, we feel the pain. That's called chronic pain. For example, some neuropathic pain. So for this type of pain, for especially chronic pain in the current medical system, we don't have a 100% medical treatment to treat the chronic pain, such as neuropathic pain. So understanding how we feel the pain, how it is happening in our body is very, very important to develop future drugs to treat pain and other related associated symptoms, disorders. Earlier, this has been mystery for 1,000 years, how we perceive the sense of pain. So in 17th century, a French philosopher called René Descartes, he made this illustration. In this illustration, he draw a thread, a long thread, which is connecting different parts of our body, and it connects to our brain. So what happens is, how he imagined was, when you keep your leg on the fire, so that is moving particles on the fire, which travels through this thread, and then it pulls the thread and it opens a valve in our brain. So that's how we sense the pain and the fire. That's how he thought. But later on, there are, in the past century, in the decades, so many basic scientific research has unraveled and discovered that it's a specialized sensory neuron, which has a long process called nerves, are present in our body. This is present all over our body, in my skin, my muscles, everywhere. And as you see, one of the nice specialized thing about these nerve cells are, they are very long. So if it has to start from your toe to your spinal cord and in the dorsal region, and some nerves can go up to your brain. So when you take a human cell, which is like few micrometer, very smaller in tiny size, but the nerve cells are specialized one, which is, if, for example, if a person is 6.5 feet long, he might have a 5.5 feet long nerve cell in his body. So I mean, if I cut this is spinal cord, if you look at the cross section of the spinal cord, you'll have the dorsal root ganglion, where you have different types of neural cells. So our whole body has majorly 3 different types of neurons, which is motor neuron and relay neuron and sensory neuron. So sensory neuron is the one which we are interested in, which Nobel laureates were interested in, to look at how the temperature and touch senses is happening in our body. And one more thing is, our nerve cells are not as our normal cells, which we read in the textbook, like not a round with a nucleus. You see the structure of the nerve cells, this is how they look like. So it's different from the normal human cell. And this is the list of all the Nobel prizes in physiology or medicine given in the past for the discovery of nervous system and other associated stimuli and studies. So the question is, how our nervous or system detect this stimuli, that is touch and heat. And the scientists thought that there must be this neuron, sensory neuron, is actually discovered taking the physical stimuli from external, like the touch and the temperature, to our central nervous system, to our brain. So there must be something at this, how it is detecting. Okay, the sensory neurons are transferring the message, but how it is detecting it, the physical stimuli or the chemical stimuli or the touch, how it is detecting. So then they thought that is a molecule which is present on this sensory neurons, which detects and converts this heat and touch as a nerve impulse, which in turn sends to our brain. So these are called molecular receptors, which is present on the membrane of nerve cells. So I'll give very briefly about this molecular receptors. See, these molecular receptors are actually proteins. So we have so much of proteins in our body. And these are all one specialized group of protein called integral membrane proteins. So why they are integral membrane? Because all the cells are covered with the membrane, as you see this lipid bilayer membrane. So this lipid bilayer membrane is very, very sophisticated, and it's very tough. It won't allow anything inside the cell. It's protecting the cell from the external environment. But these proteins, like these molecular receptors are also called proteins, are buried in those membranes. And these channels, one type is the ion channels. They have a channel actually like this. And then if there is a need, then these channels will open. And if there is no need, then it will close. This is the function of these proteins. So it makes actually a pore on the surface of the cell. And if there is a need, if you can imagine as a switch. So when you want to turn on the light, you can turn on this switch and it opens like that. If you don't want the light, you turn it off like that and closes. So this is how these proteins functions on the membrane and allow the restricted entry of ions and other molecules across the impermeable cell membrane to inside the cell. And these proteins are very, very selective for their ions or some of the ligand molecule. So these proteins, based on their function and structure, they're classified majorly into 4 different groups. One is called ligand-gated channel. So the ligand-gated channels are, there are some chemical molecules present internally as well as externally. These small molecules comes and bind to these proteins and activates it. So then it's called ligand-gated channel. And some of them are mechanically gated channels. Some of the pressure, external pressure or internal pressure activates these channels to function across the membrane. And some of them are voltage gated and some of them are always open, which means that it allows some amount of ions to go out and some amount of ions to come in so that it maintains the cellular homeostasis. So this is about the molecular receptors, which we have been talking, which we are expecting to be on the surface of neurons, sensory neurons, which detect the physical stimuli, and it takes the information into further to our brain. So our first laureate, David Julius, he was so, as I mentioned to you in the beginning, that he's so keen on his the natural products. So he chose chili. Capsaicin is an active component in the chili. That is the one which causes the spiciness when we eat the chili. So he thought that, okay, how this spiciness, that heat, how we are identifying, sensing this. So that should be a molecular receptor, which is present on the nervous system, which senses this. So he wanted to identify what is that molecular receptor. It has been shown that, as I mentioned, this sensory neuron is actually responsible when you eat the chili to transfer the information to our brain. Then it actually tricks our brain saying that, oh, there is some change in temperature in your body. And that's what sometimes if you eat very spicy food, you get the feeling of sweating because our brain start to think that there is a change in temperature. So we need to reduce the temperature, body temperature. So it gives a signal to sweat. So, but what it is? So exactly this receptor molecule, what is located? So he started a very high-risk project along with his postdoc, Michael Katerina. What he did was he first took the sensory neuron. So some of the, when you treat with the capsaicin molecule with chili, some of the genes are activated. Many genes are activated within the sensory neuron. And he took like thousands of DNA, small DNA fragments. Each fragment is one gene, and each gene can produce one whole protein. So like that, he selected many genes, hoping that one day I will find out the particular gene or which is responsible for the production of protein, which detects this capsaicin, the chili pepper's spiciness. So he did it. And he taken in the cell, which is insensitive to chili heat. And then he added this capsaicin molecule and then DNA fragments by fragments, and he measured the activity. So one day, the Eureka moment, they discovered the channel protein, which is located on the sensory neuron, and they called it novel protein, called it as transient receptor protein, V1, because capsaicin is a vanilloid kind of compound. So they call this vanilloid compound binding receptor. So it's a TRPV1. It's interesting. So now he knows that how we feel the chili spice because of this protein, the molecular receptor present on our neuron, which senses it. Okay, this is spice. And then that causes membrane potential depolarization. And that in turn tells the information. Fine. So now he discovered this protein, and then they wanted to study further. So what else this protein can do? So they did an experiment. It's a very sophisticated experiment where they heat this protein. So what happens was when they heat it, this protein, this channel got activated again. Okay, so they concluded this TRPV1 protein or ion channel is not only activated by capsaicin, but also activated by heat. So that's how he said that, okay, that's how we feel the heat of the coffee or heat of the food or heat, the fire. So this is the protein, TRPV1, TRPV1 ion channel is responsible, is present in our body, helps us to sense the heat. So the next question, okay, so we sense the heat with this protein. How do we sense a cold? Is it the same protein or it's a different protein? So David asked this question. And then what he, as I mentioned again, he is so much into the natural substances, natural compounds. And he identified an array of compounds. And finally, he came up with the mint. As you know, if you eat the menthol, some mint candy, it gives a cold feeling in our tongue. How do we get that? And if we, for example, Vicks, our normal application, if we apply on our skin, it gives the cold feeling. How do we feel that one? So again, by repeating different screening varieties of genes, and he identified there is another protein called TRPM8, TRPM8 channel, which is present in our cell, that is responsible to detect the cold in our environment. So by and Ardem Patapoutian, the another laureate of this year, also discovered TRPM8. They did it independently in two different laboratories. So with this information, yeah, apart from these two, TRPV1 and TRPM8, so researchers around the world, as well as from David Julius group, as well as Ardem group, discovered other proteins. So for example, at different temperature, if it is around 20 degrees, then this TRPM8 will activate and then respond, sense it. If it is around 30 to 40, then it is TRPM2, that's yet another ion channel, which senses that temperature. If it is above 40, then it is TRPV1. So like that, we have many transporters, many small proteins in our body, which allow us to sense different temperature. So these channels acting together, they're acting as a molecular thermometer. So we have a thermometer in our body. So we never ever have noticed that. Our body can sense even 2-degree difference because of the different channels, proteins, which are sensing the temperature. And this is one of the wonderful experiments done by Julius lab. What they did is, as we all know this TRPM8 is a protein which is responsible to detect to sense the cold. So they take the mice and they prepared 2 different chambers, and one chamber is with 20 degrees. The other chamber is 30 degrees. So what they did was, in mouse, it has this TRPM8 protein. So if this protein is present, the mouse can detect the coldness. So you can see this video. So the TRPM8 is present, and here it is 20 degrees, and here is this 30 degrees. And you see the mouse don't want to go to the 20-degree compartment because it doesn't like this cold. It wants to be warm and cozy, and it just stays only in the 30-degree compartment. It don't want to go to the 20 degree. So it says that it can sense the cold. So that's what I don't want. And what they did, they knocked out. They just removed this gene, this protein from the mouse system. And then they see, because now the mouse doesn't have this TRPM8 protein which senses the cold. And we'll see what happens. Is the mouse going to sense the cold or not? And now this mouse doesn't have this TRPM8. And you see here, it can easily go into their 20-degree chamber because it is not sensing the cold. It's not cold for the mouse. So this is a very, very clear study, shows that this cold and heat activated channels are present in our body, which responds to different temperature. And how it looks really in our body, because We know that this is a channel. What is the structure of it? So this then David Julius group discovered the structure of this protein by cryo-electron microscopy, and cryo-electron microscopy is another Nobel Prize winning technology in 2017 has been awarded. And this protein, the TRPV1 protein, which senses the heat, looks like this. It has this transmembrane alpha helices on the membrane. This is the extracellular, and this is the inside the cell, which has some other soluble proteins. And this is they identified in the presence of capsaicin, in the absence of capsaicin, and how the capsaicin molecule comes and binds and how it causes the conformational changes, and everything has been very well studied. And I'm not going deep into that information. When David Julius was interested to look for the temperature receptors, Ardem, the other Nobel laureate of this year, was interested to study the mechanosensitive receptors present in our body. So what are these mechanosensitive receptors? So these are all the receptors also present on our neuron, which senses the mechanical stimulation. If somebody touched me or if somebody hit me, how do I know that? Somebody hit me on your face. How do I know that? So mechanical stimulation, how that is being sensed to my brain. So this system has been studied in E. coli bacteria, in frogs, in rat, but unfortunately there is no study was performed in mammals. So that's what our second Nobel laureate, Ardem Patapoutian, was interested in. So his strategy was, first, he searched for the cell which has intrinsically mechanosensitivity. So he took the cells in the lab, and then he just literally poked the cell with a needle, just poked it. At the same time, he measured the activity. So he applied a mechanical force by poking it. And if at all there are some ion channels present on the cell that will react, that in turn will respond to the activity. It's a current fluctuation, that is the activity measurement. So that was his approach to detect which gene or protein is present in our body responds to this mechanical stimulation. So he identified the cell line, and then he selected 72 gene candidates with his postdoc, Bernard Coste. 72, he has been testing gene one by one. So this is all like a very time-consuming work has been done. One by one in the cell line, in the particular cell, he just silenced one gene and poked it, measured the activity. Silence, poke, measure. Like that, until 71 candidate, he hasn't get any positive response. But one day, the 72nd candidate got the result. When he poked, there is a change in the current, happens. And then they identified this protein called Piezo1. So how did they name the Piezo1? Piezo is the word derived from the Greek word PSZ, which means pressure. So there are some instruments called piezoelectric sensor. They convert different physical force into to generate a voltage. So like that, these channels which are present on the membrane, which converts the physical, the mechanical force into a nerve impulse. Electrical impulse sends to our brain. So that's what they call it as piezo channels. They named it. And in one of the talk, Ardem mentioned that it just took a few minutes to explain the work, but it really took around a year and a half to complete this task, to complete this experiment. So for the discovery. So the basic research is time consuming, but it's a mystery unraveling thing in the basic research scientific field. So once they identified the piezo channel, and that is the piezo channel which was responsible for our touch and proprioception. So when I give a warm hug and the punch, so everything is detected by these piezo channels. First, they identified Piezo1, and based on a sequence similarity, they identified Piezo2, and Piezo2 is the main one which is responsible for the touch and proprioception in our body. So Ardem also wanted to look at the structure of this, and they solved again by cryo-electron microscopy. I'm not going into the detail of the structural information, but as you can see, it has a channel, this spore in the middle of this protein. That is allowing the ions to flow inside the cell, which causes this signal to the brain. And he is the postdoc Kei Satomi was involved in solving the structure from Ardem's lab. And it has like a distinct propeller type of arrangement with the 3 different domains. And it looks like that if you view it from the membrane, from the perpendicular to the membrane plane. So the next question is, what is the role of these piezo channels? As I said, it is involved in the touch and proprioception. So then further on, they did an experiment. They removed this piezo channels in mice, and they looked at the mice lack the feeling of touch, and it cannot orient itself in the space. It cannot walk. And they also observed in some of the humans where they have deficiency in this Piezo2 channels. And that is one study where they have a picture, a video showing that a human who are deficient of this Piezo2 protein, they cannot walk properly. So that's how, if we go to the doctor with some problem in our limbs, not able to walk and balance, then it's usually been misdiagnosed as some motor neuron disease and related to it. But it's actually very specific to this piezo channel, Piezo2 deficiency. So discovery of this piezo channel will help to develop a new drug specifically targeting these channels. And there is a girl, Shweta Murthy from Ardem's lab, and she was interested to look at what is the role of this Piezo2 in a chronic pain. It's called one type of chronic pain is called mechanical allodynia. For example, we take the chilli. If we have the chilli rubbed on our skin, and it causes like a very severe pain, even in that region, if I touch it like very gently, it's paining a lot when it is actually applied. So how these piezo proteins are playing a role there. So that's what she was interested in. And she studied that in mice as well as in humans. And they show that only in the presence of piezo is mediated this mechanically induced allodynia. And that was a very interesting one. Nice work done by Shweta. I'm not going deep into that as well because it's more technical information. And so they studied initially after discovering the Piezo2 in, they showed us in somatosensation where touch, proprioception, and the pain, these piezo channels are involved. And they also started to study in other systems like in respiration, baroreflex, and bladder. For example, in respiration, when we breathe in, and then at some point we have to breathe out. So that reflex is Herring-Brewer response, which is present in our body, which makes it after breathe in, you have to breathe out. The reflex is happening. But these piezo channels are actually playing a role there. So when you breathe in, the channels are activated, and that in turn causes the signal, okay, you have to breathe out. If they mutated, if they deleted these piezo channels, the person inhale or the mouse model cannot exhale and no breathe. And that was an interesting study done with Ardem. And in case of bladder and stomach, and they are interested to look at, are there any other piezo channel? For example, we eat. As we eat, our stomach stretch and stretch. How much it can stretch? How much you can eat? How do you know the feeling that, okay, it's enough, my stomach is full, we say. How do we get the feeling? Are there any other, it's like a mechanical because our stomach is stretching. Say, for example, in our bladder, we get urine accumulated. How do we get the feeling? Are there any other piezo channels are located on these organs? How it is sensing? So Ardem is working on that. And I will talk about this baroreflex is one another interesting feature. Baroreflex is a mechanism in our body. Suddenly, if I get up, sometimes, okay, I get like feeling of giddiness. It's because sometimes if you sit for a long time and suddenly you wake up, your blood pressure goes up. And after some time, it comes to normal. So that is this baroreflex. It tells us when the blood pressure goes up and these channels, these piezo channels present in the blood vessel get activated, and it tells the heart to rate slow, and the heart rate goes down, and then the blood pressure comes to normal. So this is how the piezo channels are present in our body, and it is also present in different tissues like the sensory neurons, as we discuss now, and the inner ear and the blood vessels. Everywhere it is present, and it is giving and making our system to function normal. And this is one another example where the piezo channel is lacking. So there is a mutation in the piezo1 that actually causes this lymphedema. So usually our lymphatic system, we have the lymph fluid in our body, so the lymphatic system will hardly function not to let this lymph fluids to get accumulated in the foot, or so it just pulls up. But these piezo channels present on the blood vessels will help activate. If this comes up, down, activates, and then that in turn helps to go back. But if this piezo channel is not present and that activation is not working, then there is a condition called lymphedema happens in a patient's deficiency of piezo1 channel. And another example is an interesting one in red blood cells. Our red blood cells also has this piezo channels, piezo1. So we have seen, like, if there is a problem, if there is a deficiency in piezo, then it causes some disease condition. We can't walk or we can't feel the touch. It's a loss of function. But in this case, it is gain of function. I'll tell you why. So we have a piezo channels present in the red blood cells. Plasmodium falciparum, it's a malarial parasite, which comes once the person is affected with a malarial parasite. It goes into our liver cells and then to our red blood cells. That's where it grows. And then once it grows inside the blood cells, and then it breaks the bloods, RBCs, ruptured the RBCs for the larva to release. So that in turn causes the cerebral malaria. So in this case, there is a mutation, single mutation in the glutamic acid 756, because this protein is a very huge protein, the piezo1. It has like 2,500 amino acids, very huge one. So, but there is only one mutation, which is at glutamic acid 756, causes the red blood cell is dehydrated. It's not as healthy. So in many are having this mutation, but it's not that much genetic effect happens. So what happens is when this piezo1 channel mutation happens, the red blood cells are not very healthy, a bit dehydrated. In this case, they treat it with the Plasmodium falciparum parasite, but you don't want to go into this shrunken, dehydrated red blood cells. And then the mouse model has been protected with a malarial disease, malaria. So they also have tested this. Surprisingly, they chose the African population, those who have in America, African Americans, and they tested it because where the malaria was prevalent. Where you see the wild type, as it is, the piezo1 is present, and the RBCs are healthy, no problem with this, and then the Plasmodium will come and infect, and you get the high Plasmodium falciparum infection. But this mutated individuals, where they have the mutation, have dehydrated RBC, but the Plasmodium doesn't enter or infect the red blood cells, and that in turn leads to very low Plasmodium falciparum infection. So this is one gain of function, and this opens the path for the drug discovery of malaria, which is one of the challenging disease in the current scenario. So with this, I would like to conclude my talk that these two Nobel laureates, David Julius and Ardem Patapoutian, discovered a molecular channel which is present in our body, responsible to feel the sense of temperature and touch. So they have unraveled one of nature's mystery. And like that, so many mysteries are in our body, which has not been unraveled, and it's being done in the world around. So thank you very much for listening. And I would be happy to take questions.
Yeah, thank you, Madhu. Nice talk. Yeah, it's open for questions. I think there are some questions. Vijayan,
we can also invite the audience to raise their hands if they want to ask the question directly to the speaker.
Yeah, once Santosh has asked a question.
Yeah. Now, let me start with my question. So I see the structure of TRPV1. I didn't see the, I want to see the channel it has been creating or inside that. No, like you have shown the channel in the Piezo2, but
Yeah,
TRPV1,
exactly.
I didn't find that.
Okay,
Where is that?
it does has that one. I didn't keep that picture, but I can show you in a moment. So it was in one of David Julius. One minute. So this is the paper which describes the channel. See here, they have this channel picture, but it is to get a better visualization. As you see here, this is the whole molecule. Can you see my screen now?
Yeah.
Yeah, okay. So this is the whole molecule, and if it's a membrane plane and it is rotated 45 degree, and if you view it from the 90 degree from the membrane, and this is the pore actually caused by this trip V1. So, and if you go through and if you view it on, this is perpendicular to the membrane plane, the view, and if you go into the parallel to the membrane plane, you see this hole in the middle of the protein which passed through the membrane.
Okay. My second question with regard to the channel is, how do you quantify it? If the opening or the channel diameter is related to the intensity of the pain or the intensity of the heat or something like that?
So usually there is a technique, sophisticated technique called electrophysiology, where you purify the protein and then you take artificially created liposomes, like a lipid bilayer liposomes, and then they insert the protein, reconstitute the protein into these lipid liposomes. So it looks like the protein is in the membrane channel, and then they induce the stimuli, and that in turn releases the ions, which means these channels activation means it lets the ions to get inside. In this case, it's a cation. Some of the sodium and the calciums get inside. So when the ions goes inside, and that ion, it changes the membrane potential, the ion current. So then they connected with an electrode. They measured the change in the electric current. So that's how they detect the activation of these channels.
But it's not showing the actual temperature. If the temperature increases, the number of ions going through it will increase. Is that right?
The temperature increases, the channel has to open. So for example, if it is from 25, you start to heat, only at the range of 40, the channel starts to open. Once the channel is open, the ion starts to come in and it causes the depolarization of the membrane. Then the activation happens. Then more ions leads to the increase in the current.
Okay. It's more or less linear from the 45 to 40 degree range.
Yes. So this is for the TRPV1, and for the TRPM8, it starts when it is 25.
Okay. So now questions start pouring in. Let us go to the audience questions. Let me open that chat box.
Jay, there is a question in the chat box.
Yeah.
Charles.
First, it's from Dr. Santosh. Is there any remedy? Is there a remedy for patients who suffer from CAPA disorder, who generally suffer due to lack of pain and sweat? If the loss of touch is due to Piezo2, how these anomalies could be diagnosed, since the predominant doctors would be totally ignorant of these factors?
Yeah, that is what, like, now they have to include this as a marker. So after the discovery of this piezo channel, which is responsible for the touch, so they have to include in our medical system to screen for this gene. So when they diagnose it, they do a set of screening to identify where the problem is. So they have to include this gene screening to whether it is present or not. If that gene is absent, then the physical problem related to the neuronal problem could be because of the lack of these channels. But I'm not aware whether this screen has been implemented in the regular medical practice or not, but might not be, because it's just now this, 10 years back it's been discovered. So it might come in future.
That's another question from Mr. Ramanujam. Is this sensing or touching and temperature quantitative as well? Does the structure suggest a measure too?
Quantitative in terms of electrophysiological measurements, as well as they did calcium imaging, how much calcium comes in. But in terms of structure, it's not very qualitative or quantitative, I would say. So from the structure, you get the mechanism, how it is performing. I mean, actually, they exactly don't know how this heat activates this channel. So they know they can give an explanation how the capsaicin molecule binds and how it causes the conformational changes on the protein, but they couldn't get, even for the touch, how exactly it causes or that transfers the signal with respect to the protein structure. It's not been known both for heat as well as for the touch.
Another question is it is from Mitish Sendil Kumar. Now he's asking, dear sir, can you please help me with the slide presentation to show and explain my son along with the madam's wonderful explanation? I think he's asking the slide to be shared. He wants to see it. I don't know what is.
I know it might be there with in the YouTube.
Yeah, YouTube, it's going to be there, right?
Yeah.
The next question is, thank you very much. My question is a general one. In the course of the lecture, you talk about switching off protein like TRP amide by manipulating the corresponding gene in mice for purpose of experiment, where whether all the myriads of the protein can be mapped to a single gene. That's whether anyone one-to-one corresponding between proteins and gene.
One-to-one corresponding. If I understand the question correctly, is it possible to do these type of experiment for all the gene, or I'm sorry, I didn't get the question. I didn't understand the question. Can you please repeat the question once again?
No, the question is whether the myriads of protein can be mapped to a single gene. That's whether one-to-one correspondence between proteins and genes.
Yes, actually, one gene is responsible for producing one protein in our body. So like that, the TRPM8 channel corresponds to one single gene. So though it exists like in the protein structure as 3 or 4 molecules, but it is actually only one gene which produces one protein, one large, and then it assembles into a multiple protein. So to answer the question, yes, it is possible to map a single gene for that particular protein.
Aditya Verma, did you understand it? Are your question is addressed? If not, can you ask the question straight away? You can raise your hand and you can speak to this.
Yeah, I'm Aditya Verma. It is addressed. It is addressed.
Yeah. Okay.
Basically, I want to know whether any one-to-one correspondence is there, or
yes,
one gene is responsible for, or one-to-many, whether it is a one-to-many or one-to-one.
In some cases, in some systems, one gene can corresponds to other proteins, different protein, but there might be some differences between, in terms of the fold, how it is folded, the structural differences are there. The same gene can produce different, but how it is folded and how it is oriented in the cell makes it differentiate.
So next question from Akshay. He's asking, what's the difference between Piezo1 and Piezo2 receptors?
Piezo1 and Piezo2 in terms of structure, or when they first identified the gene or the protein, which is the ion channel from the 72nd gene candidate, was Piezo1 actually. And then they looked at the dorsal root ganglion. It's not, the Piezo1 is not expressed in there. It's only Piezo2 protein that was expressed. So they identified the Piezo2 based on the sequence similar homology, and then they identified Piezo2. But in terms of course, there is a difference in the sequence and the structural difference between these two, Piezo1 and Piezo channel, as well as in function. So Piezo1 Piezo2 is mainly involved in the touch and the proprioception, but not Piezo1. When they knocked out and they looked at, and also in the experiment with the respiration. So there, both the proteins are involved. When they knocked out Piezo1, the respiration is not affected, and Piezo2 is not. But when they knocked out both, and then they found that the respiration is affected. So it does has the differences between these two proteins in terms of sequence as well as the structural and function.
So next question is from again from Aditya Verma. He says now whether the pain receptors can be consciously controlled.
Pain receptors can consciously be controlled. I don't know. Sorry. Consciously. I mean, this is the question which was related to my my husband asked, like, when we tickle somebody, so can we control our smile? Okay, I'm tickling you, but I control my smile and I don't smile it even though I tickle you. How does it happen? Is it related to this touch receptors? I really don't know the answer. Maybe some neurobiologists would be able to answer, because there are so many factors, neurons involved in our actions, the tickling and the smiling. So, but to be honest, I really don't know the exact answer. Maybe SK can help me to answer this question if he has some idea.
Madam, my question was whether by training, I mean, mentally by training, whether these pain receptors can be changed at a particular location or
No.
Or a body automatically, I mean, adjust to the pain.
Yes, but if you say the location where it is expressed, where it is present inside our body in the cell, that is cannot be changed for sure. So once it is synthesized, it's targeted to the membrane, and that's where it is present. So the inherent nature of these protein to be targeted, because the amino acid composition, the building block is made in such a way that once it is produced inside the cell, it has to go to the membrane. So is that what you mean? It cannot change its location within our body.
My question is based on somewhere I read that there are some experiments done by giving some counseling or training to the patients without anesthesia. The operations have been done. That is my basis of this question.
Is there, yeah, it's a question. Connection is this any connection between the action of the proteins and the mind?
Yeah, I think what is being referred to is the fact that these channels only allow the ions to come, and but how it is registered with the brain and whether the brain is going to issue other instructions to the motor neurons to not perform any action. That is a different thing, because it's like you can, if the brain issues instructions to other neurons, then you may not move away your hand even though it's very hot. So this sort of also these channels have a saturation. So it is not infinite. Beyond a limit, that's a maximum that can come in. So once the channels are saturated, you will not have any further input. So that's the reason why if you keep on putting a hand in the cold water, you stop feeling it. And there's a relative temperature difference that you sense, not the absolute one, really. So I think the other question related to the brain is really not to do with the channel function. It's to do with the brain function, that if you train your brain, obviously you can prevent the brain from issuing instructions to certain parts of your body and so on. So you can prevent a reaction to pain and prevent reaction to touch and so on.
I hope he got the answer.
Two more questions. The next question is now, so ma'am, can we say Piezo2 is found only in sensory neurons and Piezo1 in other cells?
No. So both are found in sensory neurons, but the expression in dorsal root ganglion, the Piezo2 was present mainly. But both are present in the sensory neuron.
So another question from Saji. He's asking, how pain relieving bombs relieve pain?
It's sensitized, I think. It's basically not relieving the pain. Actually, it causes heat. When you apply the pain relieving bomb, it actually produces the heat, and that heat or the pain, which is actually overcome, which we sense more. So then that is dominated, actually. That is what my understanding.
Yeah, maybe another way to tell it is like an optical illusion. It's a illusion that the brain is diverted to some other activity, and therefore it doesn't really register.
Yeah, it does. Pain, it's a thing like it doesn't relieve the pain, actually.
தமிழில் கேள்வி கேட்க விரும்புபவர்கள் தமிழில் கேட்கலாம்.
நான் தமிழ்ல நான் சொல்லுவேன்.
ஏன்னா கொஞ்சம் பேர் வந்து தமிழ்ல எப்படி சொல்றதுன்னு தெரியாம இருப்பாங்க. தமிழ்ல கேட்கணும்னா கேட்கலாம். நீங்க
கேட்கலாம்.
So I have one more question. This is with regard to the pain. When somebody is kicked, now he get the pain. So how can he continue to get the pain after the kick is being withdrawn? That means now the channel is permanently open.
But how the pain? No, it will close. It will not be activated. That's like the switch. I said, no, the molecular switch. When there is a stimuli comes in, it opens up. And then it gives a signal. When the stimuli is not there, it closes back. As you see in this picture, when the mechanical force is applied, then the channel opens and then the ion flows in. Then you get the information. When there is no mechanical force, it is closed.
Then how can the pain continue to be persisting?
It's in case of chronic pain, you mean?
Yeah. So the channel, once the channel is closed, the signal is withdrawn. No, you don't have the sense of pain, right?
No. I think you will have the channels still open, because even the mild touch, you feel the pain in chronic pain.
Okay. So I think the problem is in chronic pain, we don't know why the channels are getting opened when actually there is not a thing. So that's a sort of a mismatch in that. I think that's where when it becomes very sensitive, why is there excess? There's the relationship between the amount of pressure applied and the amount of ions or the sensitivity that is caused. That is really the issue here. and that's not as studied well so far.
Yeah, and in one of their experiment also they have some basal level of expression for that, which they are not able to completely address that. Yeah.
The question from Akshay again: can a dehydrated RBC still transport oxygen molecule?
Yeah, I think so. Yeah, it can, because when they measured, they look at the population, people, those who have this allele, they are still nice. I mean, there is no any phenotypic or any changes in their day-to-day life, and it still can transport the oxygen. It's not a complete destruction, a complete dehydration.
Okay.
But it's something which is not optimal for the Plasmodium falciparum parasite to live inside the RBCs.
And Saji has asked another question: are pain channels always open in case of fibromyalgia?
I haven't, I don't know. I haven't read about that one, so I don't know whether they have done any research on that with respect to these pain channels. I'm sorry, maybe I can check that later and then I get back to you.
Okay. I think all questions are over. So my appeal to the audience: you can become a member of our Tamil Nadu Science Forum PSL group by you can share your email ID, as well as you can become a member of our Telegram group. You can get the we will send you the information through these two media. So then my second appeal is, please fill up the feedback form. I provided the link in the chat box. Please click the form and fill up the feedback. That will help us to upgrade our way of conducting the programs. It was very much helpful in the past as well. Please fill up the forms and post it immediately. You can share. I also appeal you to share your email ID and phone number. And when you are filling up the form, you can fill up the email ID and phone number. That we will pick it up and I will add you in the group. So with that, let me ask Chella Kumar to give the vote of thanks.
Yes, sir. Okay. வணக்கம் நண்பர்களே. இந்த ஆண்டு வழங்கப்பட்ட உடற்கூறியல் அல்லது மருத்துவத்திற்கான நோபல் பரிசு குறித்த அற்புதமான உரையை டாக்டர் மதுரநாயகி துளசிங்கம் அவர்கள் மிகவும் தெள்ளத் தெளிவாக நமக்கு வழங்கினார்கள். அவர்களுக்கு தமிழ்நாடு அறிவியல் இயக்கத்தின் சார்பாக நன்றியை தெரிவித்துக் கொள்கிறேன். அடுத்ததாக தமிழ்நாடு அறிவியல் இயக்கத்தையும் இந்த PSL தொடர் பற்றியும் அறிமுகம் செய்து இந்த உயர் அறிவியல் தொடரை ஒழுங்கமைப்பு செய்து கொண்டிருக்கின்ற நமது திரு விஜயன் அவர்களுக்கும், இன்றைய உரையாளரை அறிமுகப்படுத்திய மதுரை காமராஜர் பல்கலைக்கழகத்தின் முன்னாள் பேராசிரியர் டாக்டர் எஸ் கிருஷ்ணசாமி அவர்களுக்கும் நன்றியை தெரிவித்துக் கொள்கிறேன். மேலும் இந்த நிகழ்ச்சியில் எங்களோடு இணைந்து எங்களை வழிநடத்தி சென்று கொண்டிருக்கின்ற பேராசிரியர் TR கோவிந்தராஜன் அவர்களுக்கும் நன்றியை தெரிவித்துக் கொள்கிறேன். அத்தோடு பின்னணியில் இருந்து இந்த உரைத்தொடருக்கு உதவி செய்து கொண்டிருக்கும் திரு சுந்தர் உட்பட PSL செயற்குழு உறுப்பினர்கள் அனைவருக்கும் நன்றியை தெரிவித்துக் கொள்கிறேன். இதோடு இந்த நிகழ்ச்சியில் பங்கேற்று பயனடைந்து கொண்டிருக்கின்ற மாணவர்கள், பேராசிரியர்கள் மற்றும் அறிவியல் ஆர்வலர்கள் அத்துணை பேருக்கும் எங்களது நன்றியை தெரிவித்துக் கொள்கிறேன். நீங்கள் ஏற்கனவே சொன்னது போல PSL-இன் வாட்ஸ்அப் குரூப், டெலிகிராம் குழுவில் இணைந்து எங்களுடைய நிகழ்ச்சியை பற்றிய விவரங்களை தெரிந்து கொள்ளுங்கள். மீண்டும் அடுத்த நிகழ்ச்சியை நாம் சந்திக்கலாம்.
6ஆம் தேதி அடுத்த நிகழ்ச்சி 6ஆம் தேதி வேதியியல் பற்றிய Nobel Prize in Chemistry. That's going to happen on 6th of November.
Okay, sir.
Thank you.
Thank you, sir. Thank you, ma'am.
Thank you, madam. It was really a nice talk. Thank you very much.
Thank you.